Ryan Smith from TruDiagnostics. I am so excited to be here with you today because we often get to chat, but sometimes it's you asking me questions. So today I get to ask you questions about TruDiagnostics and the future of epigenetics. So before we jump in, for those listening, maybe you can just give us like a really nice summary of how do we understand what is this world of epigenetics and why it's going to be valuable to anyone at all.
Yeah, definitely. So epi just means above, so above the genome. So we sort of, I guess, start our work where you leave off, right?
That's right.
And so we're measuring all the changes to gene expression that can happen and trying to read those patterns. And the reason it's important is obviously as every cell goes from a pluripotent stem cell to the cell type that it becomes, whether it's your skin, heart, or brain, all those epigenetic signatures are different. That's how we have our cell identity and how all of our different cells behave like they should. And so there's a lot of different things that epigenetics can do, but that's really where we're picking up and trying to read that information. But it can be very tough sometimes.
So I always say that I was really glad that I got into genetics and not epigenetics, because when I listen to you do presentations at conferences, I'm like, okay, that's like a lot more complicated. But how do you see this relationship? You know, you and I often discuss that often the world sees like it's an either or relationship of genetics and epigenetics. How do you see it?
Oh, I definitely don't think it's an either or. I think it's a both situation. And because I think we're ultimately looking at different things. We're looking at a current status of gene expression, but unless we know the gene, that still oftentimes is not very relevant. So we can read certain patterns. For instance, even with epigenetics now, we can predict proteins, but if we don't control for the genetic polymorphism to those proteins, we can't read it accurately. So for us, that data combined is absolutely something that we need, but they all tell us a different story. I think I often liken the epigenetics to the software component of a computer, things that—
And we're the hardware.
Yeah, hardware, exactly. And just like, you need to know how much RAM you have, you need to know your GPUs, that's all the genetics, but how that software program is optimizing the different parts of that hardware, that's really what we cover.
Yeah. Well, I use a different metaphor, and you can steal it from me. Is that I don't mind, but I talk about insight and action. So I say like, genetics gives us insight into who we are, like what we inherited and kind of what we bring to the world. And then epigenetics is really the action that's happening. It's what's playing out in the world. And then, of course, everything we do, diet, lifestyle, environment, supplements, all of that, is kind of changing how that manifests in the world. And that's why I love the conversation of insight, epigenetics, and then action. All right, what does the future look like? What are you most excited about in your space?
So I'm really excited about, I think, that changeable component that we're able to do. I think that being able to look at before and afters on interventions is certainly helpful, so we can decide the best therapy for different health statuses. Obviously, we do a lot in aging, and so most of the research we've done has been looking at what helps to reverse the biological aging process. But I'm really excited, I think, in the future, and again, another way that our areas will combine is the combination of methylation risk scores and polygenic risk scores.
Well, let's start, because not everyone's going to understand that, right?
Yeah.
So I'll do polygenic risk scores, and then you do methylation.
That sounds great.
And then we'll bring them together. So when we talk about a polygenic or total genotype score, which is one thing, and we talk about a methylation score. So from our side at 3x4, when we talk about a polygenic score, we think about what are those kind of metabolic functions or biochemical functions that are happening, which genes are kind of impacting that function, what genetic variation is changing the way that metabolic function might be happening. And if we can create a score around that, we can get some really good insight into how that is impacting the body or the way the body works, right? So that's my piece of the puzzle.
Yeah.
Now tell us, what do you mean by a methylation polygen score?
Absolutely. So I think maybe I've talked about epigenetics, but to speak more about DNA methylation, just generally, these are the chemical tags that are sort of the off switch for our DNA. So the more methylation we have, the less that gene is active. And so we can read those patterns. That's really what we're measuring, is the amount of methylation on multiple genes, and using algorithms to determine if that's going to increase disease risk or decrease it. And so we're, again, picking up off of those polygenic risk scores, but we're reading more about environmental signatures. We're looking at diet, lifestyle, medications, and how they're affecting risk. And so what I'm really excited about is the creation of methylation risk scores, but we know that the methylation risk scores from previous work are going to be significantly better if they're paired with a polygenic risk score. And I think that just goes back to tell you ultimately why you need both. You're getting two different signals, and together they're more predictive of outcomes. And if we really want to measure and predict disease, then you need both data sets.
Right.
Yeah.
And this is why we love working with TruDiagnostic. All right. Well, Ryan Smith, thanks so much for joining us here today. And we look forward to working with you for many, many years.
Thanks so much. Appreciate it.